Good morning. I'm Lou Bridges, Physician-in-Chief and Chair of the Department of Medicine at the Hospital for Special Surgery and President of the Rheumatology Research Foundation. So I'm very pleased to be with you today, present at Room Now Live. I want to thank Jack and Artie for giving me the opportunity to present today. So the topic I'll have today is epigenetics and rheumatoid arthritis, a primer for clinicians. I have no relevant disclosures. So the outline of what I'll talk about today is, first of all, what is epigenetics? And what are some of the common epigenetic mechanisms that regulate gene expression? What is known about the role of epigenetics in rheumatoid arthritis? And can epigenetic studies in rheumatoid arthritis lead to new drug targets and ultimately novel therapeutics? So first, what is epigenetics? So how is it different from genetics? Genetics is the nucleotide sequence from a gene that provides the blueprint for a protein. Genetic mutations or polymorphisms may change the structure or function of that protein. An example is sickle cell anemia. So in sickle cell anemia, there's a missense mutation shown in the red letters here. And this is in the beta globin gene. This leads to a change in the RNA sequence, shown here as a uracil. This then leads to a change in the protein. The amino acid that is encoded for here is valine. This leads to a structural change in the hemoglobin molecule so that it clumps, and that results in the sickling of red blood cells. So this is a genetic single mutation example of a human disease. Epigenetics is, epi comes from the Greek word upon or on or over. And so really what epigenetics means is influences upon genetics. So epigenetics is a study of how behaviors and environment, such as diet, exercise, smoking, exposure to chemicals, drugs, etc., cause changes in the way that genes work. So epigenetic changes are reversible, and unlike the DNA sequence, which is irreversible, these genetic changes can be altered. Epigenetic changes can be altered. And in a simplified way to think about it is epigenetics affects gene expression, and it turns genes on or off so that they can have variable degrees of influence in human traits and in human diseases. So comparing disease states to controls might help to understand disease and potentially lead to new ways to diagnose or treat these human diseases. So next, what are some of the common epigenetic mechanisms that regulate gene expression? So this is a complex landscape, and I'll highlight a few of the common variants that are common mechanisms that are often studied in epigenetic analyses. So I'll go into these in a little bit more detail, but they generally include DNA modifications, such as methylation or hydroxymethylation, or addition of a methyl group to an adenine here. There are other ones as well. The second general category is chromatin modifications. Chromatin is the highly condensed DNA in human chromosomes. So the DNA and the histones around it. So there can be variations in the histone variants or modifications in them. A third way is through the presence of what's called non-coding RNAs that can degrade messenger RNA or somehow affect the turning on and off through direct mechanisms. And then finally, RNA modifications, which I will not spend much time on today. So with regard to DNA modifications, there are methylations groups that can be added. And so the way to think about this is a DNA methylation group that is added onto specific DNA. It blocks the proteins that go on and latch onto the DNA to read it and therefore transcribe it. And these methyl groups rather can be removed through a process called demethylation. And in general, methylation turns genes off, makes them inaccessible to be transcribed, and demethylation turns them on. So here are some examples shown here. So DNA can be modified at the cytosine C or adenine A residues by the addition of chemical groups, and cytosines specifically can be modified by methylation, hydroxymethylation, formylation, carboxylation, and adenines are typically modified by methylation. And most of what I'll talk about today is methylation. A second epigenetic mechanism is modification of the chromatin itself. So this is a picture of a nucleosome. The nucleosome is 146 base pairs of DNA wrapped around a histone octamer. So here are some histones with subcomponents histone 2A, 2B, 3, and 4. So this octamer is here. The DNA is wound around it. And when it's tightly wound, it is not accessible to be transcribed. So there are ways that the nucleosome can change position so that a particular genetic region is open up for expression, or there can be the modifications similar to what we talked about before, or variants. And this is what the structure looks like from a more molecular point of view. You see the double helix here, which is the DNA, and then here are the histones, 2A, 2B, 3, and 4. So this is an important variation that we'll talk about as well. So often in the literature, we see things referred to as histone marks. What are marks? They're tails that are post-translationally modified or marked by the balance of these writers and erasers that have an influence on what genes are turned on and off. So genome-wide localization of histone markers occurs at distinct regions, often in promoter regions, which turn on the gene itself, or enhancers, which turn on a distant gene or the gene bodies. And they often correlate with different functional impacts on transcription through the series of enzymes, in this case, polymerase II, which helps to transcribe the gene. These different histone marks recruit specific readers important for each stage of the transcription cycle. And so there's a nomenclature for these different histone marks, and that's sort of delineated here. So a mark called H3K4ME3, which is very cryptic sounding, is not that complicated when you understand the decoding. If you have your Dick Tracy decoder ring, histone 3, this means that in histone 3, there's a lysine amino acid residue, abbreviated as K, at position 4 in the histone sequence, histone 3 sequence, and it has 3 methyl groups on it, Me3. This actually is a signal to initiate transcription. There's another mark called H3K9AC. Again, histone 3, lysine at position 9, is acetylated, and this is for pause or release, so it's a downregulation of gene expression. And so on. So you can see here that there's a signal for elongation, which is trimethylation at lysine position 36 in histone 3, and for termination, which is 2 methyl groups at position 9. And so you can see here what these methyl groups look like. Here's a normal lysine residue. Here's 1 methyl group added, 2 methyl groups added, and a trimethyl lysine here. So and there's interplay between these particular modifications too that can regulate the transitions between turning on and turning off. So for example, H3K4 methyl 3 can trigger H3K9 acetylated. Another mechanism of epigenetic influence on gene expression is through a set of unrelated, what we call non-coding RNAs, which are free-floating and do not necessarily free-floating within the cell, and do not encode a protein. So they are there, and they can influence how much of a gene is expressed, because some of them have silencing complexes or represent silencing complexes, so they can actually stop transcription. these long non-coding RNAs can actually inhibit the protein expression or gene expression too. The final one that's a major part of epigenetic mechanisms is alterations to the RNA once it's transcribed. And I won't talk about that today because it has not been studied as much as these other mechanisms, particularly in rheumatoid arthritis. But there are over 160 chemical modifications that can occur to messenger RNA, as shown here. So this is meant to sort of summarize what we're talking about here. This represents a gene graphically represents a gene that is repressed or turned off. So the transcription start site is where the protein complex, the polymerase complex that leads to transcription, where it binds and starts transcribing. But here it's turned off, and it's turned off because you can see that there's methyl groups here. So there's DNA methylation shown here. And then you can see here two histone marks delineated here on the nucleosome. And the one in green actually causes pause or release of transcription proteins. And the one in yellow downregulates nearby gene expression. So this is a site that's turned off. This is a site that is turned on and is open for gene expression when there's binding of the initiation complex to the transcription start site. And it's as shown here, these cytosines are not methylated, so there's nothing to block here. And these particular histone marks, the red triangles actually promote elongation, and the gray triangles initiate transcription. So this is an example by which studying DNA methylation and histone marks, one can determine whether a gene is repressed or turned on. So this is from this figure is from a paper that I'm going to talk about in a couple of minutes, but it's a good example of some of the assays that are used in epigenetic studies. So a frequently used method is called ATAC-seq. What is ATAC-seq? ATAC-seq stands for Assay for Transposase Accessible Chromatin with High Throughput Sequencing. And what that does is it looks for areas of the chromatin that are either accessible or not accessible, open or closed. Another frequently used assay is called ChIP-seq, which is chromatin immunoprecipitation followed by high throughput sequencing. And that is a way to look at histone modifications in the chromosomal DNA. And then there's WGBS, whole genome bisulfite sequencing, which quantitates the DNA methylation level throughout the whole genome. So these are just a few of the assays that are used to understand epigenetic mechanisms and how they are working in humans, either diseased or control, or when examining a particular trait. So the next topic is what is known about the role of epigenetics in rheumatoid arthritis. So a lot of work was done by Stefan Guy at University of Zurich, who has done a tremendous amount of work in this area. And I want to acknowledge his work and summarize here by showing some of his work that summarizes his work on rheumatoid arthritis synovial fibroblast, or sometimes called fibroblast-like synoviocytes. So these synoviocytes tend to be activated in rheumatoid arthritis. There's inhibition of apoptosis and leading to hyperplasia of the synovial lining layer. There's production of cytokines and chemokines, which results in inflammation, and production of matrix metalloproteinases, which lead to tissue invasion. And some of Professor Guy's work has shown that some of these factors here, joint destruction, et cetera, that are manifestations of RA, are due often to epigenetic mechanisms that regulate gene expression in synovial fibroblasts. And he has particularly found evidence of differential DNA methylation, histone acetylation, histone methylation, and also histone summoylation, which we have not talked about, but it's a very similar chemical process, just a different chemical group is added. Instead of methyl, it's a somoyl group. So this is going again back to the study from Gary Firestein and his colleagues, which I showed you the examples of the assays a few minutes ago. So what he did was he and his group studied the, they looked at RA synovial fibroblast, RA fibroblast-like synoviocytes from both RA and as controls osteoarthritis. And so they did a comprehensive set of assays on these cells, including ATAC-seq, ChIP-seq. They also did RNA sequencing so that they would have a measure of what genes were expressed, and that would allow them to then correlate what they found here with the expression levels found in these synovial fibroblasts. And they also did the whole genome bisulfite sequencing. So here's a representative example, and I want to walk you through this very complicated slide, but this is an example of the relative signal intensity across a particular region of the genome for the histone marks in rheumatoid arthritis and osteoarthritis fibroblast-like synoviocytes. So up at the top here, you have the chromosomal region. So this is chromosome 11. It's displayed linearly, and you can see the different parts of the chromosome shown here. Also in purple are the summary of the rheumatoid arthritis fibroblast data, and in green are the osteoarthritis fibroblast data. The genes that are in this region are just depicted by vertical lines shown here. And they studied 6 histone marks, H3K27AC, and you can see the other 5 here. And they designated each by a different color. They also looked at open chromatin, the RNA sequence for expression, the DNA methylation across the genome, and they found, pardon me, they show here, really, this is a representative example. It does not show any significant differences, but it's meant to show how generally reproducible these results are. So this is the results of looking at this histone mark in RA synovial cell fibroblasts and in OA. And you can see here there are some differences, but I'm not going to discuss this in detail, but I have one representative example on the next slide of where there are some differences. So you can see here in this particular chromosomal region shown here, if you look at RA on the top and OA on the bottom, these do appear to be significantly different. And what this is a graph showing is at H3K27AC, you can see the number of differentially modified epigenetic regions is high when you compare RA to OA. And you can see that graphically in a more geographic design along the chromosome here. And again, this is RA and OA. So this gives clues as to what regions may be transcribed in RA compared to OA. So interestingly, when they did this study, which was reported in Nature Communications in 2018, when they compared RA to OA, they found some very interesting things. And one of the most interesting was they found the Huntington's disease signaling pathway was overrepresented or overexpressed in RA compared to OA. And this is shown with a specific gene called HIP1, Huntington interacting protein 1. So this was completely unexpected. And so this is an example of how studying epigenetic influences can lead to interesting new observations. And so when they studied this further, they found that the Huntington's disease signaling pathway was the most commonly enriched pathway among their RA samples, but it's not been known to be associated with immune-mediated disease previously. The HIP1 appeared in many locations, HIP1 participates in endocytosis of proteins from outside the cell into the cell, and cell signaling and cell invasion in cancer. And they found that the HIP1 deficiency, when they knocked it down with interfering RNAs, it unexpectedly reduced the invasion of the phenotype of the fibroblast-like synovial cells by nearly 50%. And when they looked at the shape of the fibroblasts that were deficient in HIP1, they changed shape from this normal structure, normal shape of a fibroblast, which is spread out, to one which is much more condensed and has a great deal of difficulty migrating, suggesting that HIP1 plays a role in invasion into cartilage in RA. And this was found through these epigenetic studies. Firestein and his colleagues have also studied differentially methylated pathways between peripheral blood and synovial T cells in RA and in OA. So this is a set of comparisons of RA peripheral blood methylation in T cells versus methylation in RA synovial T cells. And you can see that there are several pathways that seem to be very different, rather, in blood compared to synovium, such as pathways involved in the innate immune system, integrin cell surface interactions, complement pathway, P50 pathway, and TNF alpha signaling through NF-kappa B. They also found a number of differences between RA peripheral blood and OA peripheral blood, as listed here. They surprisingly did not find a tremendous number of differences between RA synovial T cells and OA synovial T cells, and these are really more metabolic pathways and not immune pathways. So this was interesting. So to sum this paper up, they analyzed DNA methylation of CD3 positive T cells, which is a pan T cell marker, between peripheral blood and synovial tissue in RA and OA. They found 4,615 in differentially methylated loci and 164 in RA and OA samples, respectively, resulting in 832 and 36 differentially methylated genes. So the loci that they found that were differentially methylated, a small subset of them, or a relatively small subset of them, resulted in differential methylation of the gene compared to the overall loci. And they saw that methylation differences in T cells were greater on the basis of location, blood versus synovium, more than they were, surprisingly, at least to me, than they were between disease process, RA versus OA. And again, their insights led to potential better understanding of the pathogenesis of RA and maybe potentially gene targets, drug targets rather. So the differentially modified pathways were significantly enriched between RA and blood and synovium, especially in a set of genes related to complement, integrin cell surface interactions, and P53. So their conclusion from this was that the patterns of DNA methylation in RA show location-specific differences in immune pathways, whereas methylation differences in OA are limited. And they speculate that the RA joint-specific signatures could be due to selective accumulation of T-cell populations or to expansion of differentially marked adaptive immune cells, such as B cells or others. And understanding epigenetic patterns could provide clues to the types of T cells that accumulate in the RA joint and could lead to identification of therapeutic targets. So I want to make a brief mention of microRNAs, because there have been quite a few studies looking at microRNAs as potential diagnostic or prognostic markers in RA. So microRNAs are small, usually 17 to 25 nucleotide pieces of non-coding RNA that are floating around, and they regulate gene expression by either repressing translation or causing degradation of target mRNAs. These microRNAs are thought to play an important role in the development of the immune system and in regulation of immunity. And more than 100 microRNAs have been identified that could affect molecular pathways in development or function. And shown here are a few of the microRNAs that have been studied and found to be reported as potential diagnostic or prognostic markers in RA. So for example, microRNA 16 has been studied in synovial fluid and shown to be increased in fluid compared to other autoimmune diseases. And you can see further down the line, the types of samples are analyzed, including synovial fluid, plasma, whole blood, PBMCs, et cetera. And you can see the different microRNAs that are thought to potentially be associated with the presence or absence of RA, with the progression or lack of progression or less progression of RA, and then also potentially some that may be markers of treatment response to TNF inhibitor, B-cell drug, methotrexate, anti-TNF, or others shown here. These are the microRNAs, and here's the tissues or the location from where they were studied and the associations that were found. So finally, I want to just sum up with a few words on, can genetic studies in RA lead to new drug targets and ultimately new therapeutics? So we've talked about epigenetic mechanisms on the chromatin. We've seen this here, but these can lead to health endpoints such as cancer, autoimmune disease, mental disorders, diabetes. These have all been implicated through one way or another through epigenetic factors. And among the most active compounds under clinical development are histone deacetylases. So these histone acetylase transferases and deacetylases, as well as protein methyltransferases, et cetera, are emerging. So the histone deacetylases are abbreviated HDACs, and these remove acetyl marks on histones, and they're organized into 5 families shown here with the different numbers of the different individual enzymes shown here. So these proteins have been shown to play a role both in cancer and in human development, and they can target classes or they can be specific for individual HDAC proteins. And they're being evaluated as both monotherapy as well as combination therapy for a large number of diseases. So shown on this slide are some FDA-approved HDACs, or HDAC inhibitors, I should say, like belinostat, which targets these HDACs and is now approved for peripheral T-cell lymphoma. And you can see other approvals for multiple myeloma, myelodysplastic syndrome, cutaneous T-cell lymphoma as well. And so these are just some representative examples. So a quick recap: epigenetics is a study of how behaviors and environment can cause changes that affect the way genes work. There's a complex landscape of epigenetic mechanisms, including DNA modifications, chromatin modifications, and non-coding RNAs. Epigenetic studies in RA have led to the discovery of novel pathways, such as the Huntington's disease signaling pathway, and better understanding of gene regulation in blood compared to synovium. MicroRNAs may play an important role in the pathogenesis of RA, and epigenetic studies of RA and other diseases may lead to new drug targets and ultimately novel therapeutics and better ways to treat or prevent rheumatoid arthritis. So I thank you very much for your time and attention. My contact information is here, and if there are questions or you'd like to discuss any of these things in the future, please feel free to reach out to me.